Hello everyone. When you were marking your questions, the answers that you wrote, I asked you for your marks and then I said, look, are there any questions that you want me to explain? So I'm just going to spend about 5 or so minutes going through the questions that some people highlighted. So you'll need your questions and answers in front of you. And start off just looking very quickly at the first part to question 1. So on this, the key thing is that we're looking at mitosis. You know that mitosis, the daughter cells are genetically identical to each other, but they're also exactly the same as the parent cell. So you were asked to draw the daughter cells that would be produced if the top cell divided by mitosis. So they're going to be exactly the same. Now let me see. So you've got one like that. You've got one like that. Short one, another one of those bent ones, one over there, and one over there. Now, strictly speaking, they don't have to be arranged in the same way, but they both must have the same chromosomes as the parent cell. So that's what they were looking for. The examiner would check that we've got the correct shapes of chromosomes and the correct number of chromosomes, and the same as the parent cell. Down at the bottom there, asexual reproduction isn't really something we've talked about, but some plants will produce asexually. So strawberry plants will, whenever their stem bends over, touches the ground, a new plant will start to grow. Or there are other plants that will, maybe your mum or dad has been visiting somebody and they've liked a plant, and the person says, well, look, I'll cut a little slip of it for you and you can take it away and grow your own. And they maybe just cut a little side shoot off and give it to your mum or dad. They take it home, put it in a jam pot full of water. It starts to grow roots. And then after a few weeks, your mum or dad plants it in soil and it grows into a new plant. So that's asexual reproduction. There's no gametes, there's no male, female involved there. But thankfully the question isn't asking you about that. It's basically saying, how is mitosis used in living organisms apart from reproduction and cloning? So things like growth and repair, or to replace worn out cells. Question 2E showed us part of a DNA strand, and it said, explain the effect of a random change in the DNA molecule at position X from base C to G. So if we had T, G, A, if you look along there, well, T, G, A, we get amino acid 1. So instead of having amino acid 3, you would have another amino acid 1. So you get 1 mark for saying that amino acid 3 it changes or is replaced by amino acid 1. Now, that means that we've got a totally different protein because we've got a different combination of amino acids. So they're going to bond in different ways. If it was an enzyme, it mightn't be the right shape, and so therefore it wouldn't work. So 1 mark for saying that you've got amino acid 1 instead of amino acid 3. 1 mark for saying you get a different protein being formed, or if you said the protein mightn't function properly. This change is called a mutation, and that really was the next bit. Question 3B gives us a lot of data and talks about Chargaff. Now, you don't need to know who Chargaff is, but they might just give you a question saying, look, a researcher called Edwin Chargaff did this. This is the data he got. What conclusions could be drawn? Well, if we look at this, you have an idea about these bases. You know that A binds onto T. So if you look at that, you can see that the amount of A is more or less exactly the same. So 30.9, 29.6 is really, really close. And the same here. So you get a mark for saying that the amount of A equals the amount of T. And then also for noticing that these two are equal, G and C. Anything else that you could notice? If you notice that A has the largest amount, or that C had the least amount, or that the amount of A and T is greater than the amount of G and C. Anything else logical would have got you your 3rd mark. Yes, here's the calculation then that some of you found tricky. So sheep have 21% cytosine in their DNA. Straight away, cytosine, you know that it binds to guanine. So for every cytosine molecule, there will be a guanine molecule, so you know that guanine therefore must also be 21%. So C and G together, 21 plus 21 is 42%. What are we left with? So if we take 42 away from 100, we're left with 58%. And half of that will be adenine and half will be thymine. So half of 58, should be able to do that in your head. That's 29% is adenine and 29% is thymine. So your answer, and what percentage of adenine is there? 29%. Now, what you should do is just check. So we've got C, G, A, and T. They should all add up to 100. So let's just do the sums. 9 and 9 is 18, plus another 2 is 20. 2, 4, 6, 8, 10. That's 100%. Okay, so that's a common kind of question that you might get in a DNA question. Question 4a, part 1. The first bit is asking about the similarities, and then part 2 asks you about the differences. So let's just think about DNA. We know that DNA is made up of sugars, deoxyribose. Whoops, made a wee mistake there. Sugars and phosphates and sugars and phosphates. And we've got bases in there. Now, of bases come in. So the first question says describe the similarities in the structure of DNA from different people. So what will be the same between your DNA and my DNA? Well, this bit here, the sugar phosphate backbone. This isn't writing, hold on. Sugar phosphate backbone is going to be the same. What also is going to be the same, the adenine always pairs with thymine, or A always pairs with T, and G always pairs with C. The shape of it is a double helix. So straight away, that's 3 things that are the same. It's a double helix, so this double helix shape. Phosphate and sugar backbone are the same, and the A always pairs with T, G always pairs with C. What is different between your DNA and my DNA? Well, it's the order that the bases come. So the sequence or the order of the bases is different. Now, this is question 4, part B, just underneath. So you are asked, let me see. Use the information in the diagram to give the letter of the missing base for the first amino acid. So you're not expected to know what codes for serine, but you are expected to have the wit to look along there and say, oh, there's serine. It's A, G, C, so this must be C. Use the diagram to explain and describe and explain what effect this mutation will have. So it says, just in between there, it says a mutation occurs in the DNA strand causing the base G, circled, to be replaced by another base, A. So if we have A, G, G, are we going to get glycine? Well, let's see, do we have any A, G, Gs along here? There we go. So we're not going to get glycine, we're going to get arginine. So 1 mark if you said that glycine will be replaced by arginine. And because we've got a different sequence of amino acids, it's going to give us a different protein, so it's going to function differently. And so you can see that question has come up several times. So that's a very common question in a section of the paper. Here's another cell division question. This one here starts off looking at mitosis. And as you would expect, in this one we've got 4 chromosomes in the parent cell, stage 1. And each of the daughter cells are genetically identical, and they're identical to the original parent cell there. Just when I mentioned the word genetically identical, in the controlled assessment homework that you did and I marked there, one of the questions said compare the daughter cells, and a lot of you said they were genetically identical, and that got you the mark. Some of you said they were the same. The examiner isn't going to give you a mark if you say the same. You're expected to know that the term you use is genetically identical. So let's try and get into the habit of using that. That's just worth pointing out. So look at the diagram. Name structures A. Well, structures A are chromosomes. How does the cell change between stage 1 and 2? Well, if you look closely there, the nuclear membrane has disappeared or broken down. What happens to structures A between stages 2 and 3? Well, there's nothing drawn there, but you know, if suddenly we had 1 there and now we've got 1 in each, obviously the DNA has replicated, or each chromosome has made a copy of itself. Why are these cells here described as clones? Because they're genetically identical. The type of reproduction which uses mitosis to produce clones, well, again, not strictly on your specification, but that's asexual reproduction. The type of reproduction we studied earlier in Unit 2, where we had male and female, that's sexual reproduction. As we said, some plants will reproduce and we'll get cells like this, and this will go on and produce a new organism, and this one here will go on and produce a new organism. There's been no sexual reproduction there, no male, no female cells, no gametes, and so that's called asexual reproduction. I really don't think they're going to ask you that, or they should ask you that in an exam, but it's just making you aware of that term. Somebody had put down an answer called cytokinesis. You've obviously been looking up online about cell division. That is something that happens after mitosis or meiosis, when the cell actually splits. So it's really happening at this point here when the parent cell splits in two. That's what cytokinesis is. It's not a word you're expected to know at GCSE, but it is something you'll come across if you do AS biology. Question then was the second half of this, and this time we're talking about meiosis. And you can see there we've got 4 chromosomes in the parent cell at stage A. Daughter cells have got 2 chromosomes. So you had to complete the diagram. So down here, this cell on the left has got one big one of these and one small one. This one over here has got a white one and a small white one. So what are we going to draw in here? Well, there should be another big black one, and there should be another big white one, because remember, that will make a copy of itself. So there's one there, and there's the other one, and the same with the black one. There's one and there's the other one. In terms of the small ones, we've got a small black one. It doesn't actually matter whether you put it in that cell or the other, and a small white one. It doesn't matter how you arrange them. They could be the same as what we've got already, or it could be a mixture. It doesn't actually matter. So long as you end up using the right chromosomes. So in other words, you've got altogether out of the 4 daughter cells, you've got 2 big black ones, 2 small black ones, 2 big white ones, and 2 small white ones in any kind of combination there. This question over here. 2 differences between the daughter cells produced by mitosis and meiosis. Well, in mitosis, there are 2 daughter cells, but there are 4 daughter cells in meiosis. That's a good one to start with. In mitosis, the daughter cells are genetically identical. In meiosis, they're not. And you could say that in mitosis, the daughter cells have the same number of chromosomes as the parent cell. In meiosis, they have half the number of chromosomes as the parent cell. And the word for that is diploid and haploid. So if you said in mitosis, the daughter cells are diploid, they're haploid in meiosis. And one type of cell produced by meiosis, you could have sperm, ovum or egg, or just the general term gamete. Okay, so hopefully that makes sense to you. Have a look at the controlled assessment that I marked over the weekend, and bear that in mind. Some of the words came up again today, but say spend a bit of time revising and then do your test, hand it in today or later on Monday, okay? Thank you, bye.